So hello and welcome to BioAffairs. Today I'll be teaching you about the 7 crucial, most important difference between gram-positive and gram-negative bacteria. So depending on the different aspects, they can be differentiated very well. But the most important are I have prescribed here. So start with the cell wall first. So we know that both the bacteria contain cell wall and they contain a NAM and NAG molecules, N-acetylmuramic acid and N-acetylglucosamine, as a cross-linking or cross-linked in their cell wall. The NAM molecule contains 4 amino acids which cross-link with another NAM molecules or another amino acid present in the NAM molecule, and they make a bond, right? And that is the difference between the two bacteria, gram-positive and gram-negative bacteria. How the cross-link happens in those both the gram-positive and gram-negative bacteria. So NAM molecule contains L-alanine, D-glutamine, then L-lysine or mesodiaminopimulic acid, and at last there is D-alanine. So these are the 4 amino acids. Now the variation is in the third position. So at the third position of tetrapeptide side chain, tetrapeptide means 4 amino acids. There, in case of gram-positive, L-lysine will be present. Okay, and in case of gram-negative, there will be mesodiaminopimulic acid, or sometimes abbreviated as BPA, is present. So that is the crucial difference between them. And a pentaglycine bridge is present in case of gram-positive cell wall. So pentaglycine, 5 residues or 5 molecules of glycine is present between the chain which is interlinked. So try to understand that the 4 amino acids, that chain is forming from D-alanine and connected to another NAM molecule's third position. So that in this, let me draw a picture, it will be easier for you. So suppose this is a NAM molecule and this is a NAM molecule. Now position 1, 2, 3 and 4. Here also position 1, 2, 3 and 4. So D-alanine is present in the 4th position. So from this 4th position, 3rd position is connected, right? So the 4th amino acid is connected to the 3rd amino acid of another NAM molecule. Likewise, the cross-linking happens. Now in this bridge between the 4th molecule and the 3rd molecule, here what happens? Pentaglycine. So 5 glycine molecules may be present in case of gram-positive cell wall, which is absent in case of gram-negative cell wall. So these two is a very major difference between the gram-positive and gram-negative bacteria. Now this is a general difference where the gram-positive cell wall is much more thicker, that is 16 to 80 nm, compared to gram-negative cell wall that is very thin. As you can see, it is only 2 nanometer compared to 16 to 18 nanometer. So it is around, what I can say, it is largely suppressed in case of gram-negative. Now coming to the lipid content. So the lipid content is very scanty or scarce in case of gram-positive bacteria. Whereas in case of gram-negative bacteria, there is a high number of lipid content is present in the form of outer membrane, the structure present in the gram-negative bacteria outside the cell wall, or including cell wall, whatever you say, it is outer membrane, and this is mostly composed of lipid. Okay, so high lipid content in case of gram-negative bacteria. Now the lipopolysaccharide layer or the LPS layer, that is totally absent as it do not contain any outer membrane or any lipid containing structure. So it is absent in LPS. LPS layer is totally absent, whereas in case of gram-negative, lipopolysaccharide is present, which works as an endotoxin. So it behaves as an endotoxin if there are bacterial infection inside the body, human body. Okay, so this will function as endotoxin. Now teichoic acid, it is exclusively found in the gram-positive bacteria. It cannot be found in the gram-negative bacteria. So where it is present? It is present in the cell wall. Sometimes it combines itself with lipid and form lipoteichoic acid, which is exclusively present in the gram-positive cell wall. So as you can see, most of the difference present in the gram-positive or gram-negative cell wall. So both these characteristics makes them very much different in their behavior depending on the cell wall composition or cell wall structures. The next is variety of amino acid. So the variety of amino acids in case of gram-positive cell wall is very few. Means similar type of amino acids are present in their all the cell wall structure. Whereas in case of gram-negative cell wall, there are several kinds of amino acids can be found, which makes the need more pathogenic or diversified antigenic response because of the presence of several amino acids, different amino acids. Now aromatic amino acids, it is totally absent in case of gram-positive cell wall, whereas in case of gram-negative cell wall, there are presence of aromatic amino acid. Okay. Now the last is the resistance to antibiotics. So gram-positive is very much susceptible to antibiotics, whereas gram-negative is somewhat resistant. This also signifies the importance of cell wall because it has a thick peptidoglycan layer and it is not covered by any kind of outer membrane or lipid membrane. What happens when antibiotic is applied around the environment, microenvironment where the bacteria is present, that time bacteria attracts or the bacteria attaches to the antibiotic or engulfs the antibiotic very easily, and it is easily going inside the gram-positive bacteria because of the presence of a thicker peptidoglycan layer and the absence of any kind of LPS layer or outer membrane structure. Whereas in case of gram-negative bacteria, the presence of lipid molecules or LPS layer or outer membrane makes the antibiotic much more reduced in containing them inside the body. So that's how it makes them more resistant compared to gram-positive bacteria. So as you can see, most of the characteristics belong to the cell wall differences. So gram-positive and gram-negative bacterial difference you can describe from the cell wall itself. Okay, that's the major differences, 7 crucial differences between the gram-positive and gram-negative cell wall. As you can apply it to gram-positive and gram-negative bacteria also. An extra one point you can add with the examples. You can give some examples about gram-positive bacteria and gram-negative bacteria. Okay, so I hope this will help you in your exam. Thank you very much. If my classes are really helping you in your studies and it is really clearing your concept, and if you really like my classes, give a big thumbs up and don't forget to subscribe my channel BioAffairs.